Last class we were discussing about characteristics of plant cells, then how are they different from microbial fermentations. So what impact bioreactor different operating parameters would have in plant cell fermentations. So something which is peculiar to plant cell fermentations, we spoke about cell aggregation, which is more frequent in plant cell fermentations, then the foaming and also the wall growth. Then we spoke about mixing, where we need radial as well as axial mixing, and I also said that in plant cell fermentations, because they are heavy, more dense as the cell density increases, so suspension efficiency has to be enough and is an important aspect which has to be taken care apart from dispersion of the gas bubbles or the mixing or homogeneity inside the reactor. So for which the impeller design is crucial. So generally, if you remember, I said for plant cell fermentations, high width to diameter ratios are preferred, and moreover the distance between the sparger and the impeller and the kind of spargers, like for example point spargers, if they will be used, then they will have more instability in terms of there is higher chance, lesser dispersion possible. But with more radial mixing, dispersion improves, but then there is a demerit to suspension, axial movement. So therefore helical ribbon or centric impellers, centrifugal impellers, these are specific impeller designs which are frequently used for plant cell fermentations, because they can create both axial and radial movements and better KLA in comparison to used impellers like radial impellers for microbial fermentations, and less shear forces on the cells. So pneumatically agitated reactors, mixing is a limitation, but obviously because there are no moving parts, so shear rates are less.
What is pneumatically agitated reactor?
Pneumatically agitated means air driven, there are no moving parts. Whatever mixing is being provided is provided by the airflow rate, sparging. So dissolved oxygen levels should be maintained above the critical oxygen concentration. Now high culture viscosity, cell shear sensitivity, cell aggregation, all these phenomena make oxygen transfer in plant cell cultures a challenging problem. So their impeller design and sparger design, as we were talking about, will help in overcoming these problems. Now factors which will affect the KLA, which is mass transfer efficiency in the reactors, which will include superficial gas velocity, culture viscosity and reactor geometry. So when we say on basis of what that I spoke about that these factors will be affecting KLA, there will be, you will find in literature enough empirical correlationships between mass transfer characteristics, how they are related to various operating parameters of the reactors, for example impeller width, impeller diameter, then gas superficial gas flow velocity, power input to the impellers. So these are some of the parameters in the reactors which have been found to be related with the mass transfer coefficient of that reactor vessel. So why plant cells in comparison to microbial fermentations are less shear tolerant? Because see, there is an inverse relationship that when the plant cells are in the early log phase rapidly multiplying, then they are more flexible. But as the cell density would increase, they become lesser flexible to shear forces. So reactor configurations which you will see in literature, these are all pneumatically driven reactors. The first one is your stirred tank reactor. Now number of impellers also can be one of the factors for mixing, the amount of mixing which is needed, which is quantified in terms of mixing time. Then in the pneumatically driven reactors, you will find there are tapered bubble column reactors, then there will be airlift reactors, external internal loop reactors. All these, now knowing that these are pneumatically driven reactors, what is the limitation of such kind of reactors? Mixing is a limitation. So any changes like making an external loop to it or making it tapered in the design is improving the mixing time or trying to overcome the limitation of the pneumatically driven reactors. The position of the sparger, where the sparger should be placed in an airlift of a bubble column will also determine whether there can be dead zones at the bottom or not. If the, like for example, I will show you a picture, it will come later, where you will see in literature they did a study where the position of the sparger was changed, and they could find that in an inner loop reactor, when the position of the sparger is well above the loop, the draft tube, then the circulation currents were better because the bubbles could drive the currents from inside to outside. But because if the sparger was placed well below the draft tube, then there is a chance that there would be dead zones created because the bubbles will get dispersed at the bottom rather than all the bubbles flowing through that channel, creating density gradient and circulation currents. So, let us talk about the different configurations. Now, stirred tank reactor is a well known configuration which is very well used for microbial fermentations. Now, stirred tank reactor, the difference which you will find in plant cell fermentations is with respect to the sparger and the impeller design, absence and presence of baffles. Now, even people are working out with the aspect ratio. Now, the kind of reactors which I was talking about, for example, disposable bioreactors where or wave bioreactors, they will have more surface to volume ratio. Now, the kind of impellers like anchor impeller, propeller, 6 flat bed turbine, this is well used for microbial fermentations. Now, why these designs? Because these designs, as I said, will impact your flooding transitions or flooding and loading. Now, this impacts the dispersion of the gas bubbles in the reactors. More is the dispersion of the gas bubbles, better is the mixing, but there is a limitation of creating suspension of the cells. So, sparger, whether you should use point spargers, whether you should use a ring sparger also depends on the reactor geometry. So, all these aspects have to be taken into account when you are designing reactors. Now, alternative designs which you will see, these are disc turbine curved blade reactors, then large diameter impellers. Airlift reactors, these are pneumatically driven reactors. So, the first picture is your inner loop reactor, the second is the outer loop reactor. Now, outer loop reactor which will improve mass transfer. Why? It is said that the outer loop improved mass transfer of oxygen, gas liquid mass transfer was improved. Because
air is being sparged and that's why.
In both the cases air is being sparged.
How does that external loop work?
It is a simple enclosure which is in continuity with the inner reactor. So, what is increasing? You are having an extra path. So, what? So, you are giving more time for gas hold up. More the time, more is the time given for exchange to happen. So, more circulation time. Then this is what I was talking about, the position of the sparger if you see. So, when you want effective mass transfer to happen in pneumatically driven reactors, you want the suspension also to happen, mixing with suspension. So, what is causing this suspension and mixing? It is only the circulation currents. And what is causing the circulation currents? The density gradient. What is the cause for the density gradient? The gas sparging. So, if you can see in the picture, it is very well demonstrated that if you change the position of the sparger a little up in the draft tube, then the gas bubbles would drive in the direction in which it is desired, thereby creating the right circulation currents and improving mixing. So, lesser chance for dead zones at the bottom of the reactor. The other thing which they have shown here is that if the lower edges can be filled rather than keeping it very erect, edgy at the bottom, and they have been made circular, thereby filling it with glass, then this will improve the circulation rates of the medium, avoiding the dead zones at the bottom of the reactor. The disposable bioreactors, we have already discussed about the wave bioreactor. This is a well known bioreactor used for animal cells, but people have also started using it for plant cell fermentations. It works on the principle of wave and undertow. Wave everybody knows, undertow, it works on the principle of wave and undertow mechanism. So, why this mechanism is being spoken about? There is no moving part. So, what is causing mixing? This motion, rocking motion. Now, this rocking motion would create what? Ripples, currents. So, it will create wave that at the surface. Now, even so, the same principle what works at the beach. When the wave comes to the beach, then there is a large mass flux of the liquid towards the beach, but there is an equivalent mass flux against the beach which is beneath the trough. So, that is undertow, those currents which are backflow beneath your wave. So, they work in the opposite directions. So, this is what will cause mixing to happen when this wave gets created. So, that is what is called as wave and undertow mechanism. Slug bubble reactors, they are very high aspect ratio reactors. High aspect ratio means very high height to diameter ratio is large. So, in this such that a single bubble is created which can span the entire column, entire reactor volume as it moves slowly, and the mass transfer would take place as it would move on the edges of the bubble. It is said that it will create a plug flow. So, on the edges of the bubbles there is a very thin film of liquid which is left adjacent to the glass column or the reactor wall. So, through that the liquid and the gas transfer between the liquid and the gas takes place through that thin film which is adjacent to the bubble and the wall at the top of the bubble, if you can see on the sides of that bubble which is shown in filled grey color. So, these are called slug bubbles because they will create slug flow, slow motion, but and you can assume it closer to plug flow, no boundary layers. Now, bioreactor operating conditions, apart from choosing the right kind of configuration if you want to improve the productivity. Now, we have spoken about different strategies which you can use to improve the yield of the product or the productivity of the product. Then you, under those conditions, you choose the right kind of bioreactor, you apply those conditions, you get to some X productivity. Now, what is the limitation? Now, I need to improve further beyond this. Is it done? Is the bioprocess now optimized? No, there is still ample scope for productivity enhancement. In what way? We have learnt about different strategies. You can count now. There are n number of strategies which we have learned which can improve the product yield, secondary metabolite yield and productivity. Now, the limitation in the reactor would be a time will come when I will have to stop the reactor, be it any reactor. What will be that time? That time would be invariably when the nutrients are done. Can I do anything to never allow nutrients to be done till whatever point I want? So, that is what you can overcome with your mode of cultivation. Now, in that I will have to design which nutrient to feed and at what time to feed, at what flow rate to feed, and should I be using, suppose I optimize the nutrient composition, it came as 30 grams per liter carbon and nitrogen, and I saw that as she said secondary metabolite, I did statistical optimization and I found that carbon is the most critical nutrient for biomass, but phosphate is the highest impacting nutrient for my secondary metabolite. So, then if I want to improve the productivity, which is a sum of the biomass and the product, what should I do? I would say it is better to run in one reactor itself and have a two-stage cultivation.
We will increase the biomass, then we will change the substrate.
So, which means that what should be the composition of the feed will also be governed by what I have studied in the batch, whatever optimization I did in the shake flask. So, that is where what concentration of the feed and what composition of the feed should I be feeding in will be determined now at the reactor level, which can then further improve in the same reactor the productivity. So, that is what is bioreactor operating strategies, where you can even do it as a fed batch or a continuous cultivation in the chosen reactor configuration. Now, an effective bioreactor operating strategy provides a high productivity, a high product yield. So, we have been talking about high product yield with respect to biomass and high product yield with respect to substrate. Now, high product yield with respect to biomass will reduce my which cost? Upstream, downstream? High product yield with respect to biomass will reduce, I said, the cost. Which cost will it reduce? Upstream, downstream? Downstream. High product yield with respect to substrate will reduce my production cost. So, that is why these two factors should also be optimized, can be optimized separately. The operating strategy is determined based on the pattern of product formation. To relate the pattern of product synthesis to cell growth, appropriate index for cell growth is needed. Now, modes of cultivations which you will come across will be multi-stage batch, fed batch reactors, single and multi-stage continuous chemostats, then repeated batch which are also called as fill and draw systems. See, you will hear many different terminologies, but if you will read them, see the details, you will be able to see that there is a lot of overlap between one or more of these. Perfusion reactors are nothing but cell recycle reactors. So, chemostat with cell retention. So, cell retention devices, so they are called as perfusion reactors also, and immobilized bioreactors. Now, immobilized reactors for plant cells, what do you need? Good bulk mixing required to minimize external mass transfer limitations, so that there are lesser boundary layers around the beads or the stagnated biofilm. Adequate exposure to air or the aerated medium. So, sometimes you will see that when the culture reactor is separate from the medium. So, it is called as a reservoir and the biomass is kept in a separate reactor. If suppose it is being worked as a packed column reactor, packed bed reactor, then there is a reservoir, the media is circulated from the reservoir continuously and the spent media is again recirculated to the reservoir. So, why do you think this is being done not in one reactor? How will that help? There is a separate reservoir because it will help you to aerate the medium. Once it comes out, you separately in the reservoir, the medium is aerated such that you have enough dissolved oxygen and then it is circulated, because mass transfer is a limitation in immobilized reactor systems or the external mass transfers. Now, immobilization can also affect plant cell metabolism, so cell viability has to be tested. So, different reactor designs, they involve what? Surface attached cells or as biofilms, or these will facilitate what? Cell to cell contact, and generally you will see that as the cell to cell contacts improve, this may also impact the secondary metabolite biosynthesis. These are some of the examples in literature. People have used immobilized reactors. I was also talking about hollow fiber reactors, where either inside the tube the cells can be immobilized or outside the tube the cells can be immobilized, but again with growth there would be mass transfer limitations. So, the flow rates have to be optimized. Now, packed bed reactors, the cells are now what is the limitation of packed bed reactors? There will be higher pressure drops in packed bed reactors because there is more resistance to flow, and but that limitation is taken away in well mixed immobilized reactors. You use the cells and you make them freely suspended in solution. So, they become like reactors with cell retention. So, there the limitation of external mass transfer can be avoided. Then fluidized bed reactors, in which from the packed bed reactors there is fluid movement to improve the mass transfer, but then the only way the cells are kept suspended depends on the flow rate of the gas or the fluid flowing rather than gas, because the fluid flow rate will determine the suspension efficiency of the cell mass also. Now, if I improve the flow rate, there will be better suspension, but then there is lesser time given for the mass transfer to happen. isn't it? So, then what is done is you may see that there are circulation developed through reticulate matrices such that you drive the liquid currents or the fluid is forced to spend more time with the cells, or you will see that lot of gas sparging is done. Now, gas sparging is done in these reactors to improve the bulk mixing and to facilitate mass transfer between the gas bubbles and the liquid medium. So, that is one way of improving the mass transfer of oxygen in these reactors. But then what is the limitation? The limitation would be that you are having high gas velocities with liquid circulation currents. So, the limitation can be high amount of shear which may get generated. Membrane reactors, we have already spoken about. Hollow fiber reactors, you can go through whatever is written here. Now, despite all this, there are known limitations in plant cell fermentations. One thing is, the minute it is brought to in vitro cell lines, the yields drop. So, and moreover plant cells, we know that there is what is the limitation with plant cells? Callus. Plant cell suspensions are created from callus, isn't it? Cell lines. So, there is somaclonal variation. So, because of which the biosynthetic capacity may vary. Now, in order to avoid this, people came and exploited the agrobacterium mediated transformed cultures, especially the hairy root cultures. Why? Because they are organ cultures and heterologous expression can even be done. It will become more stable because there is genome integration of the tDNA into the plant chromosome. So, it is more stable transformation event. And there is the other limitation in plant cell fermentations, you need hormones, but in hairy root cultures the cost will come down because they do not need hormones for growth.